talk about Chapter 20. We are moving into our discussion about biology, which will be our last group of science topics. So in Chapter 20, we're going to talk about the nature of living things. And so to start off, let's talk about what it means for something to be alive. Because biology is the study of living things. So the characteristics of life are the five that are here. Metabolic processes, and this some examples are nutrition uptake, nutrient processing, waste elimination, generative processes, which is growth and reproduction, responsive processes, which is irritability, which is response to stimuli, not being irritable, individual adaptation, population adaptation, which is evolution, control processes, which is the coordination and regulation of metabolic activities, and then structural similarities of our cells. So here are some examples of those five characteristics that we talked about with the examples underneath them. This image is from your textbook, and so you should be able to view this larger if you need to. But you can see here that there's a picture example to go along with each example. So early cell theory, Anton von Leeuwenhoek, 1632 to 1723, was the first to see an individual cell, and he called them animalcules. Robert Hooke, in 1635 to 1703, studied thin slices of cork. So cork comes from a tree. You can see the image there on the right-hand side of the cork. And he termed the individual structures that you see the term cell, which is the word we use now. So two types of protoplasm were later identified: the cell nucleus, which is the central body within fluid, and then the cytoplasm, which is the fluid portion of the protoplasm. So it's outside the nucleus. So we know now there are two main types of cells. The first being your prokaryotic. These are these cells that don't have a true nucleus like we do. So they are our bacteria and our archaea. And then the second cell type is called a eukaryotic cell. This has a true nucleus as well as other membrane-bound organelles, which we're going to talk about. And the examples for that are algae, protozoa, fungi, plants, and then animals. Now, there are five common traits of all cells, no matter if they're eukaryotic or prokaryotic, and they are that they are housed within a cell membrane, that they contain a cytoplasm, which is the fluid outside of the fluid within the cell membrane. They have genetic material that's necessary for reproduction processes. There's energy transfer molecules, and then there's also enzymes and coenzymes. So let's talk about the cell membrane. The cell membrane is a thin sheet shaped into different structures and forms related to the function of the membrane. So the cell membrane is made up of phospholipids and proteins. The phospholipid is a polar molecule that forms a double-layered sheet, and we call this the phospholipid bilayer. Tells you that there's two layers in water with water-soluble portions facing away from each other. So each of these is a single unit, and there's going to be a water-loving side and then a water-fearing side. And so the membrane is going to line up to where the part that doesn't like water is going to be on the inside, and the part that likes water is going to point towards the outside and the inside. And then this is going to act as a barrier to protect the inside of our cell from things outside the cell that don't belong there. So let's talk about how things move across the membrane, because the membrane is there to protect us, but it also is going to have to allow some things to pass through. So diffusion is one of the processes we'll talk about. It's the net or overall movement of a molecule from a place of relatively high concentration to a place of low concentration. This is called a passive process because it doesn't require any energy in order for the movement to occur. So it is not going to use ATP. And ATP is going to be the energy currency of the cell. And so it's kind of like a ball rolling downhill. That's going to be the process that doesn't require any energy, but moving the ball up the hill requires energy. So the rate of diffusion is going to be related to how much energy is there, the size, and the concentration of what's moving across the membrane. So if we're looking at the image here in blue with the red dots, obviously if this was a cell, all the dots have no incentive to basically clump up there in the corner. They're instead going to separate and fill the entire blue box. If we're looking at the bottom right image, diffusion is the one there on the left where you have the blue circles just moving through. It's a passive process. And diffusion will occur until something that's called dynamic equilibrium is met, which is when the molecules are equally distributed and movement is equal in all directions. So it means if two move into the cell, then two are going to move out of the cell, and it must be balanced. There's no net movement, meaning if we have movement in one direction, there must be equal movement in the other direction, and this means that there is no concentration gradient. So diffusion is going to occur when there is a concentration gradient, meaning there's a difference of concentration on one side of the cell and the other. The second type of process we're going to talk about is osmosis, which is very similar to diffusion, except instead of solute particles moving, it's going to be specifically water moving through the selective permeable membrane separating two solutions. So passive movement was going to occur from an area of higher concentration to lower concentration, and only certain molecules can pass through based on solubility, size, and ionic charge. And osmosis is going to occur because keeping the concentration of the cell is critical. If you have too much water in a cell can dilute the contents and interfere with the chemical reactions. Too little can cause buildup of waste products. And so what cells are looking for is to exist within an isotonic solution, which is where the water and the dissolved mineral concentrations in the cell will equal the surroundings. And that's where you're going to have that dynamic equilibrium. If the cell is in a hypertonic solution, this means that there is not enough water. And if the solution is cell is in a hypotonic solution, it means there's more water. So what does this look like? Say you have a selective permeable membrane, and on the left you have a 60% water, 40% sugar, and on the right you have less sugar and more water. Well, because the sugar molecules are too big to fit through the gaps, you can see if this was like a puzzle, you wouldn't be able to fit the square through the circle. So the water is the only thing that can move. And the water on the left is less than on the right, so the water molecules are going to move from right to left to help balance out the concentration. So how are cells affected by this? Well, we talked about isotonic, hypotonic, and hypertonic. So in a hypotonic solution, we talked about the fact that there's going to be more water on the outside of the cell. So because there's more water outside the cell, water is going to rush into the cell. The cells are going to swell and eventually they'll burst. So cell death will occur. If it's a hypertonic solution, there is not enough water outside the cell. So the water is going to be forced to leave the inside of the cell, travel out into the solution, which is going to cause the cells to shrivel up. They will shrink up, and you can see both the red blood cells and the plant cells are going to shrivel on the inside. So when you get dehydrated, essentially this is what happens, is that the hypertonic solution is present and all of the cells will lose their water to compensate for that lack of fluid. So facilitated diffusion is going to be different from passive different from diffusion in only one way, and that's that it's going to require a specific protein to carry the thing across. So this would be like if you were crossing a river and you couldn't just walk through the river, but you had to take a raft or you had to take a float. You needed assistance to get across the river. These molecules that can't make it across on their own have to have a specific carrier transport protein to help them. But they're still going to move from an area of high to low. It's still passive because there's no energy required. And it's a specific carrier protein that must be present. So they can't just get on any old raft and cross the membrane. They have to get on a specific raft that's specifically designed for them. So facilitated diffusion is still going to be passive because it's diffusion, but it's going to be facilitated with a protein. So active transport, on the other hand, is going to be the opposite of diffusion. It is the constant movement of low to high. It's moving the ball up the hill again. Therefore, it's going to take energy to make that happen. The cell will expend ATP, which comes from our metabolic processes we'll talk about a little bit. And ATP is going to break down into ADP plus P and energy. It's a very specific process that occurs, and only certain molecules or ions can be moved. And again, there has to be a specific carrier protein to be present. So in that way, it's a lot like facilitated diffusion, but it's not going to be an area of high to low. It's low to high, and it requires energy. Phagocytosis is the process in which a cell will wrap around a membrane, wrap a membrane around a particle, either food, bacteria, virus, or foreign material, and basically engulf it. So it's often called cell eating because that's kind of what it looks like, is a cell opening up, making a Pac-Man around it, and then bringing it into the cell. And this is another type of way that things move across the cell membrane, but it's a specific type, and it's done by the cell membrane itself, not based on its permeability like the other ones are. Okay, so we're going to take a break here and then pick up on chapter 20 in the next video.